Okay, so in this video we will be demonstrating the validation script, which we call the validation script. This script will go through two processes. It will first crop your full-size images into small crops, small like stack of images, and then use your desired cell detection algorithms on those small images to validate whether your detection algorithm is working well or you need to tweak it a bit more. Okay, so what you first will need is your original images. Here we will be using this C7W animal data that I have on the cluster computer right now. This dataset has two channels, the 639 channel, which is the CFOS staining images, and then the 488 channel, which is the background channel. As for scripts, you will be needing, let me just delete some of the old ones. You will be needing mainly 3 files. This variable file, and then experiment parameter file, and then this detection validation jobs.sh file. This sh file we will be running at the very end to initiate this entire process, and then the variable file will contain the variables necessary for the pipeline, and then this experiment parameter file will specify where your original data is and what kind of data it is. Okay, so let's open one by one. So first we will be opening this experiment parameter demo file. You may already know, but what is very important in this file is this base directory file, base directory parameter. So this will specify where your original data is. In my case, it will be in this path. So I want to copy this path to the clipboard and then paste it here. So now it will tell the script where the data is. The other important thing will be this variable file. This will be pointing to the variable file that I will be using. In this case, it will be this file. So I will again get this copy to clipboard, include paths, and then paste it here. So now the script also knows where the variables are stored. The other things should be covered in other videos, but you want to specify which is the signal channel, which one is the background channel, and what kind of image processing method you're going to use. The option is a spot detection method or an elastic method. We will be using the spot detection method for today, but the elastic method should work in the same way. Okay. So now once that's done, let's save this file. Next, let's go to this one. Oh no, this one. Okay. So the couple tabs that you will be using will be this. The first one you will be using is this crop generating parameter tab. Okay. So the key component here is the two flags, the crop flag and the detection flag, and these other parameters. As I explained previously, this detection validation script will go to two phases. It will first generate it will crop your images into small bits, and then it will do the detection on those images. So if you put the crop flag in true, it will do the cropping. If you put the detection flag in true, you will do the detection. Right now, the images does not have any crops, so we want to first just generate the crops. So I will put this in true and the detection flag as false, and then you can specify certain other things here, how you want to generate the crops. The end crops will specify how many crops that you will generate. I will typically set this to something like 50 to avoid to get more variability of from your original images, but for today I'll just set it as 10. You can also split your crops into 2 groups. So one typical thing that people do is to separate them into test datasets and the training datasets in order to train a classifier. You want to train a classifier using the training dataset and validate that classifier using a test dataset. You can do that here by setting something like 0.1. In this case, it will generate 0.1 portion of your number of crops will be the test dataset, and then the 0.9 will be your training dataset. I will set this for 0 today. Z offsets means that you're going to just skip the first 1,000 images of your original dataset. You can just straight up go to some parts of the brain that you think there should be good signals. And then you can also specify the size of the crops. I will set it as 100 and 100 and 100 for today. Crop per stacks, you can set it as 5. This is just like, the smaller it is, you will get more variety from your original dataset. The larger it is, it will just focus on a specific region. It's not so very important, so you can just set it as 5. Okay, once that's done, save this, close this, and then go back to the scripts file. Okay, so these 2 are updated. The last one should be this one. Okay, so the things that you want to be careful about is where the experiment file. This has to be the same name as the one that you're going to be using. You have to point where the SIF file, the container file is. And then for the time, you can make it quite small. The first step will typically take like 5 to 30 minutes. So like 1 hour would be just fine. And then all of these other things will depend on your system. Okay. So that's all done. So now you want to submit this .s, the batch script. To do that, you can always log into your cluster computer. But you can, if you're using WinSCP, you can use this button to directly go into this console. You can type in bash and then batch submit detection validation jobs.sh. Then you can execute. If it goes well, you will get something like this. And then the script will generate another .sh file. If you have multiple data sets that you want to process in a batch, you will get multiple of these sh files. So what the cluster computer is doing now is it's finding a node to process the script, and this may take some time depending on how busy the cluster is. So once the script goes through and you go back to your data folder, you will see this newly generated underbar crops file folder. This will contain 2 folders, a train and test folder, and then in here you will see these new images. The best way to examine these is just to download them into your local. And then in the meantime, open image J. And then you can go into your file, then choose an image or choose all the images, and then you will get something like this. Okay. So now that you have these crops, you can go to the next step, and then you can run the cell detection part. Okay, so let's go back to the scripts folder. Okay, so now you want to go back to this variable file, open it, and then put your crop flag to false, and then instead put your detection flag to true. So now what it's gonna do is it's gonna do the cell detection. And as explained in another video, this pipeline has 2 ways to segment your cells. You can use the spot detection algorithm, which will be using these spot detection parameters, and then you can also use an elastic classifier that will have a classifier that will require a classifier pre-uploaded onto your file. folder. Okay. So for today, we're going to use a spot detection parameter. There's a bunch of parameters that you can adjust. This is a trial and error process. You can modify it and then you can check it and then modify it and check it again. There's more documentations on a GitHub page, so you can please use the link to that page. Actually, we can go here. So if you look up Clearmap, there will be this folder, there will be this documentation file, and in here you will find a lot of description of what these parameters will do. Okay, so for today I'm going to use this default set, and then close this, and then resubmit this batch submit detection script again. Okay, so once the script is done, you will again see these full files, and then inside this .out file, there will be a lot of these like random things. And at the very end, you will see the entire process ended in 20 seconds. It's actually a very fast process, normally. Okay, so you go back to your folder and you go back to this file. And then if you update this, you will actually see that now there's all sorts of new files in this folder. Let's download this again. Okay. All right. So now what you want to check is that for each file, this, for example, this train number 1 file, there will be, I guess, 6 associated files. There will be this spot refers to spot detection algorithm, and then there's this going to be cells npy file, and then cells check TIF file, TIFF file, and then cells all points npy file. Intensities npy, intensities all points npy, and an unfiltered cells check.tiff. So each of them looks complicated, but when you look at them, there's mainly cells intensities or cells all points, intensity all points, and cells check and unfiltered cells check. Basically, all points or unfiltered mean that it's not filtered yet, and then all the other ones are after they do filtrations. So what filtration means is that inside your variable file, you will find this threshold parameter tab, and here you can threshold your cells after they go through the first part of cell detections. The idea is that in spot detection or elastic, you get a bunch of cell candidates and then you narrow it down into your true cells using these thresholds. Okay, so but the first thing you want to do is examine these files. Okay, again, so open image.j, and then we can open this original file. So this is going to be the crop that we're going to investigate right now. And then you can load in, some images can read this straight up, but some can. You can use import bioformats. And then, yes, this is the one. You want to read this one. And then now you will see your raw image in red, and then you will also have a green channel, which will be showing where the cells, what the algorithm thought where the cells are. You can kind of see that a lot of cells are getting detected, but it could be getting too much. Okay, a good way to look at this in more depth is to do a maximum projection or a z-project. I would typically do something like 5 to 25 max intensities, and you can see that there's all sorts of cells that are not detected in this algorithm. Okay. So when you don't like your results, another thing you should do is to take a look at the unfiltered cells. So this will be the other file that you want to check. Okay, so this is unfiltered cells, and here you will have way more green dots than the red dots, because this is not, all of these dots are candidates, but it's not filtered at all. Again, you want to like probably do a maximum intensity projection, and you get like way more green dots. The important thing to keep an eye out right now in this process is that all of your cells that you think are, all the signals that you think a cell is, whether they have a green dot or not. If they don't, you have to at least get them to be cell candidates to do anything else. So you should probably go back to the variable file and then adjust these parameters. But in this case, what's happening is that all of these red bright cells that you think are cells do have the green dot on it. The issue is that not all the other like dimmer places also have these green dots. So this is an issue of thresholding. So what you want to do is to determine a good thresholding, threshold parameter, so that you can narrow down to the cells that you truly want to say that are cells. Okay. To do this, what you use is these NPY files. These NPY files are NumPy files. You can load this in Python using the NumPy packages. There's two types, cells and intensities. The cells will contain the coordinate of the cell. So if the cell, the green dot, if you the cell position will be, say, like x-axis 35, y-axis 45, and then z-axis like 15 or something like that. The intensities file will contain other relevant information of those cells. Those will be, for the spot detection algorithm, there will be 4. It will be the raw intensity, the intensity after a Gaussian filter, and then the intensity after you do a background subtraction part, and then the last one will be the size of the cell. If you use an elastic algorithm, you actually get different types of outputs. You will only get 2. You will get a raw intensity data, and then you will also get the size data. Okay, so what you can do is look into this file to determine what will be a good threshold. Okay, so to do this, you probably want to use Python. So I'll first get this path, and then I already have this Jupyter notebook that I use. So what you can do is load all the necessary packages, and then specify the path where you have this NPY file. So I pasted it that. So intensity array path will specify the NPY file we were talking about. Intensities all points.mpy, and then the intensity array will load that path. Okay. So today we're using spot detection. So the spot detection will have 4 parameters, as I just explained: raw intensity, DOG, that stands for the Gaussian filter intensity, background subtraction intensity, and size. You will be using this. So this entire NPY file can be converted into a data frame that will look like this. So the raw intensity from all the cells in the images will be like this, and then the intensity after the thresholding, after the filtering, will look like this, and then the intensity after background subtraction will look like this, and the size will look like this. You can further plot a histogram to see what will be the good cutoff. So this will not necessarily give you a good idea of the upper limit, but you can definitely say that there's a lot of crap in the small in the up in the lower limit. You can also just use something like mean, and then this will give you the mean or the average of each of these parameters. So from here you will start determining what will be the good thresholding and then apply it to the algorithm. So for now, what I'm gonna do is I can definitely specify it based on size. You see there's a decline here and then there's like a small bump here. So I would probably use a threshold in between somewhere here to say that anything smaller than that will not be a cell. Okay, so let's go back to the script. Let's open this variable file again. Oops, try, skip. Okay, oh yeah, I already have it open here. Yeah, so here and then I will go to this threshold parameter. And then here I will specify what I want to have as my threshold variables. What I would specify row means which type of variable I want to use. As explained here, the first one will be raw, the second one will be DOG, will be another intensity and size. I want to use size as the variable to use as for the thresholding. So I would use row 0, 1, 2, 3. So I'll be using row 3. I will specify the row as 3, 3. This is the way it's coded, so just type in, if you want to use the first row, use 0, 0. If you want to use the second row, you use 1, 1. And then for the threshold, I will probably just set it as 100. And then the upper limit, we probably want to get the maximum, or like anything above the maximum, so let's do, okay, so the max of the size is like around 200. So if you give it like 1,000, that should be probably fine. Okay. So once it's saved, you close it again, and then you can resubmit this job again. Okay. So again, this may take a while. So we wait. Okay, so once the script is done, you can check that in your file, like that. You go back to your folder and then download your files again to examine. Once that's done, you can go and open it in ImageJ again. And then, yeah, you can open these files. So after changing some of the parameters, you will get to see that there's way fewer green cells, green dots in areas that you don't think there is a cell, and it looks a lot more cleaner. Okay. So what you will be doing is you will be doing this process to all of your images until you get to a point that you're satisfied with the parameters. Ideally, what you should do is to also do a manual count and compare the automatic counts onto the manual counts. In that way, you will be able to quantify how well your spot detection worked by quantifying false positive or false negatives ratios, and from there you can also calculate an F1 score. That will be a way to quantify how well your segmentation worked compared to the true cells that you manually scored. And But once you figure out a set of parameters that you think are useful, you can use those and then save those and then run and proceed to the other script. Oops. You can proceed to this batch submit job scripts, which will do the entire full pipeline from resampling, aligning, and then segmentation of the full data set, and then transformation of those cell coordinates onto the brain atlas and things like that. But this is how the validation script is organized and what you can do using that.